In 2002, a group of researchers sat down to look at the results of the largest genetic study of human diversity ever attempted. They had spent years collecting DNA samples from over 1,000 individuals, representing 52 different populations across 7 regions of the world. Indigenous communities in the Amazon, nomadic groups in Central Asia, remote Pacific Island populations, isolated villages in the Himalayas and the Caucasus. They had sequenced millions of genetic markers per sample and built a database that, for the first time in history, allowed scientists to compare the actual DNA of populations from virtually every corner of the human world. The analysis was supposed to confirm what many scientists assumed they already understood about human variation. Instead, when the results came back, several members of the team asked the same question: is the equipment broken? Because the data did not show what anyone expected. The patterns in the DNA did not match the categories that centuries of human classification had created. The boundaries that seemed so obvious and so real in the visible world did not exist in the genetic data at the level anyone predicted. And 2 findings in particular produced a silence in the room that one researcher later described as the kind of silence that happens when something you thought you understood turns out to be something else entirely. This is the story of what scientists found when they compared the actual DNA of human populations from across the entire planet. And it is one of the most important scientific stories of the last 20 years. I am your name, and this is your channel. Subscribe now if real science is what you come here for. We do this every week. Let us get into it. The project that produced these results is called the Human Genome Diversity Project. It began in the early 1990s with a specific and, at the time, controversial goal. Its founders wanted to collect genetic samples from indigenous and geographically isolated populations before those populations disappeared into the broader genetic mixing of modern globalization. Critics raised serious concerns about consent, exploitation, and the ethics of treating human communities as biological resources. Those debates were real and important and shaped how the project eventually operated. But what the project produced, once those samples were collected and sequenced with the most advanced DNA reading technology available at the time, was a dataset that fundamentally changed our scientific understanding of what human genetic variation actually looks like. The samples came from 52 populations, spread across Africa, Europe, the Middle East, Central Asia, East Asia, Oceania, and the Americas. Each individual sample was run through sequencing processes that could read millions of genetic positions in a single analysis. The raw output filled enormous amounts of digital storage, and when the analysis software finished processing it all and the numbers came up on screen, the first thing researchers noticed was a figure that has since become one of the most cited in all of human biology. Any 2 human beings on Earth, chosen completely at random from anywhere on the planet, share approximately 99.9% of their DNA. A person from Beijing and a person from Nairobi. A person from Stockholm and a person from Lima. A person from Tokyo and a person from Lagos. Pick any 2 human beings from any 2 populations on the planet, and the genetic difference between them is, on average, about 1 letter in every 1,000 letters of the genetic code. The other 999 letters are identical. That finding alone was not entirely new by 2002. Scientists had been moving toward this understanding for years. What the Human Genome Diversity Project added was the specific detail about where that 0.1% variation actually sits, how it is distributed across populations, and what it tells us about the history of our species. And this is where the 2 findings that silenced the room come in. The first was about Africa. When the team mapped genetic diversity across all 52 populations and across all 7 geographic regions, they found that the greatest concentration of genetic variation on Earth does not lie between continents. It does not lie between the populations that look most visually different from each other. The greatest genetic diversity on Earth lies within the African continent. 2 individuals from neighboring villages in Ethiopia could, at the DNA level, be more genetically different from each other than either of them is from a randomly chosen person in Norway. That result required some careful explanation of how it makes sense, and understanding it properly is the key to understanding everything else that follows. Modern humans appeared in Africa approximately 300,000 years ago. For the vast majority of that time, our ancestors stayed in Africa. Populations spread across the continent, separated into different groups, adapted to different environments, accumulated different genetic variations, came back together, mixed, separated again. 300,000 years is an enormous amount of time for genetic diversity to accumulate. By the time any group of humans first migrated out of Africa, the African continent already contained within its populations an enormous range of genetic variation built up over hundreds of thousands of years. What traveled out of Africa was not all of that diversity. It was a fraction of it. The current scientific consensus places the major out-of-Africa migration at around 60,000 to 70,000 years ago. A relatively small group of people crossed into what is now the Middle East. and from there spread over tens of thousands of years across Asia, Europe, Australia, and eventually the Americas. And here is the crucial point. When a small group of people leave a larger population, they can only carry with them the genetic variation that exists among those specific individuals. They cannot represent the full genetic diversity of the population they left behind. Geneticists call this the founder effect. The founding group is a sample, and that sample is necessarily smaller and less varied than the original population. Here's a way to picture it. Imagine a jar containing 100 marbles of many different colors, representing all the genetic variation in an African population. Now imagine someone reaches into that jar, pulls out 15 marbles at random, and puts them in a new jar. That new jar now represents the founding population that migrated out of Africa. Even if those 15 marbles eventually multiply into billions and spread across every continent, the total range of variation in all those billions can never exceed what was in those 15 original marbles. And those 15 marbles, no matter how widely they spread, will always represent less total variation than the original jar of 100. The data confirm this prediction with remarkable precision. Genetic diversity decreased in a systematic pattern as scientists followed migration routes further from Africa. European populations showed less genetic diversity than populations in the Middle East. East Asian populations showed less than Central Asian populations. And Native American populations, representing some of the last major migrations, crossing into the Americas via a land bridge that existed across the Bering Strait during the last ice age, showed the least overall genetic diversity of any group studied. The pattern was clean, consistent, and matched exactly what the founder effect model predicted. Every migration out of Africa was a genetic bottleneck, a narrowing of the total variation carried by that founding group. All the populations living outside Africa today are, in a precise genetic sense, subsets of the variation that already existed within Africa before anyone left. Now here is the second finding, the one that produced the deeper silence. When researchers looked at the genes responsible for the physical traits that human beings have historically used to categorize each other, the traits that we visually associate with what we call race, they found that those traits represent less than 0.1% of the total human genome. Skin color, hair texture, eye shape, the features that have driven centuries of classification, discrimination, and conflict. They are encoded in a tiny fraction of our total genetic code, and they are, in evolutionary terms, extremely recent. The genetic variants responsible for light skin in European populations appeared in that population roughly 8,000 years ago. In the full span of human history, which stretches back 300,000 years, 8,000 years ago is extraordinarily recent. Blue eyes, which many people think of as a defining European feature, emerged even more recently than that. The current genetic evidence suggests that the specific mutation producing blue eye color first appeared somewhere between 6,000 and 10,000 years ago, likely in a single individual or small group living in a region around the Black Sea. Every blue-eyed person alive today can trace that specific genetic variant back to that single origin point. It is a very new development in the history of our species. The physical characteristics that human beings have used to categorize and rank each other for centuries are surface adaptations that happened in the blink of an eye compared to our full evolutionary history. They evolved because they provided advantages in specific local environments. Darker skin provides better protection against ultraviolet radiation in regions close to the equator where sunlight is intense. Lighter skin allows more efficient production of vitamin D in northern regions, where sunlight is weaker and winter days are short. The feature called the epicanthic fold, which characterizes the eye shape of many East Asian populations, may have provided protection against cold winds and glare from snow in the environments where it evolved. These are functional adaptations to local conditions. They are not markers of deep genetic difference between populations. They are recent surface-level responses to different environments. This last point connects to something biologists call convergent evolution, and it provides one of the clearest demonstrations of how misleading the visual traits are as guides to genetic relationship. Aboriginal Australian populations and populations in southern India both evolved very dark skin. But these two groups are not closely related genetically. They adapted to intense sun exposure independently through different genetic pathways because they faced similar environmental pressures in different parts of the world. The underlying DNA mutations that produced dark skin in Aboriginal Australians are not the same mutations that produced dark skin in southern India. They arrived at the same solution through different genetic routes. This is exactly what scientists see when they study dolphins and sharks, which both evolved streamlined bodies and fins, despite one being a mammal and the other a fish. Convergent evolution produces similar results from different starting points when similar environmental pressures are acting on different organisms. The same principle explains why traits do not cluster neatly along racial lines. The genetic ability to digest milk into adulthood, called lactase persistence, appears in Northern European populations, in certain East African pastoralist communities, and in some Central Asian groups. It is absent in most East Asian populations and in many African communities. The trait spread with dairy farming cultures, not with racial groups, because it provided a survival advantage specifically in populations that raised cattle for milk. Two people, classified as different races, whose ancestors both kept dairy cattle, might both carry this trait. Two people classified as the same race, whose ancestors came from different cultural and agricultural backgrounds, might not. Tibetan populations living at extreme altitudes in Central Asia have developed specific genetic variants that allow them to process oxygen more efficiently under low oxygen conditions. Andean populations in South America, living at similar altitudes on a different continent, developed a completely different set of genetic variants that achieve the same physiological result through a different molecular mechanism. Both groups can survive at elevations that cause serious health problems for lowland populations. They solve the same problem through entirely different genetic routes because they adapted independently to similar environments. Now here is the first major twist in this story, and it is one that most coverage of human genetics leaves out entirely because it complicates the picture in a direction that nobody expected. When scientists began extracting and sequencing DNA from ancient human remains, they discovered something that fundamentally changed the meaning of what it means to be human. Modern humans, at some point in their deep history, did not stay genetically separate from the other human species that were living on Earth at the same time. When modern humans spread out of Africa and into Eurasia, they encountered Neanderthals, who had been living in Europe and Western Asia for hundreds of thousands of years. And they did not simply coexist or compete. They interbred. The genetic evidence for this is now clear and replicated in multiple independent studies. Most people alive today whose ancestry traces back to Europe or Asia carry between 1% and 4% of their genome from Neanderthals. This is not contamination. It is inheritance. The genetic material from those ancient interbreeding events has been passed down through generations for tens of thousands of years and is present in billions of living people right now. But it goes further than that. A separate ancient human group called the Denisovans, known primarily from fragments of bone and teeth found in a Siberian cave, also contributed to the modern human genome. Populations in Melanesia and Aboriginal Australians carry up to 4 or 5% of their genome from Denisovan ancestors. When you look at the full picture, some modern humans today carry genetic contributions from at least 3 separate populations: anatomically modern humans, Neanderthals, and Denisovans. The concept of a single pure human lineage is not just socially constructed, it is biologically incorrect, even at the level of what we call human. Here is what makes this relevant to everything else we have discussed. The populations that carry the highest percentage of Neanderthal DNA are not African populations. They are European and Asian populations. This means that the groups that European racial science historically classified as most advanced and most civilized actually carry more genetic material from a separate species than the African populations that same racial science historically ranked lowest. The genetic data inverts the hierarchy completely. The second major twist involves ancient DNA and what it has revealed about populations that were previously assumed to be genetically stable and coherent. Modern Europeans are not descended primarily from the people who first inhabited Europe after the last ice age. Ancient DNA extracted from skeletons across Europe has revealed that the hunter-gatherer populations who lived in Europe for thousands of years after the ice retreated were largely replaced by incoming farmers who arrived from Anatolia, which is modern-day Turkey, approximately 9,000 years ago. Those Anatolian farmers did not kill or displace the hunter-gatherers in a single event. Over thousands of years, they spread, mixed, and eventually came to dominate the genetic makeup of European populations. But that was not the end of the mixing either. Around 5,000 years ago, a third wave of people called the Yamnaya, pastoralists from the Eurasian steppe, swept into Europe and contributed substantially to the European genetic profile. Modern Europeans are a three-way mixture of populations, none of which knew they were European, because Europe as a cultural category did not exist when any of this was happening. The same complexity shows up in East Asia. The Han Chinese population, which is often treated as a single monolithic racial group in popular discussions, is actually the result of a merger between northern and southern populations that mixed and unified over thousands of years of imperial expansion. The genetic diversity within China rivals the genetic diversity across the entire continent of Europe. Categorizing over 1 billion people as a single racial group obscures the enormous genetic variation that actually exists within that population. Native American genetics shows perhaps the most dramatic example of unexpected complexity. Most researchers expected Native American ancestry to trace back to a single founding migration across the Bering Land Bridge. The actual genetic picture is significantly more complicated. Some Native American populations in South America carried genetic markers that link them not only to Siberian ancestry, as expected, but also to populations related to Aboriginal Australians and Papua New Guineans. This is a genetic connection between populations separated by the entire Pacific Ocean, and the mechanism by which that connection formed, whether through an ancient coastal migration route or some other pathway, is still being actively investigated. What the data clearly shows is that human migration across the planet was far more complex, far more interconnected, and far more surprising than any simple linear model would predict. The research also produced one finding about common ancestry that has the most profound implications of all. Mathematical models based on the genetic data suggest that the most recent common ancestor of all people currently alive on Earth lived approximately 3,000 years ago. That means if you trace back through your family tree for roughly 120 generations, you will reach an individual who is a direct ancestor of every single person alive today, not a symbolic common ancestor. An actual person whose genetic material flows through every living human family line. Your family tree and the family tree of someone from anywhere else on the planet converge at that same point, just 3,000 years in the past. Going back further to approximately 7,000 years ago, every person who was alive at that time and who has any descendants alive today is an ancestor of every person currently living on Earth, every single one. The farmers in ancient Mesopotamia, the herders in the Eurasian steppe, the hunter-gatherers in southern Africa. If their family lines survive to the present, you descend from all of them. So does every other person alive. The genetic boundaries that human beings have fought and died over for centuries do not exist in the family tree of our species. They were never there. What all of this means for medicine is something researchers are still working to communicate clearly, because the implications are significant and the current practices in some areas of medicine have not yet caught up with the genetics. Some genetic variants that affect disease risk do cluster in certain populations. Sickle cell anemia is more common in people with ancestry from West Africa and other malaria endemic regions. Tay-Sachs disease occurs at higher rates in Ashkenazi Jewish populations. Cystic fibrosis affects European populations more frequently than others. These patterns are real. But the genetic analysis shows they do not mean what traditional racial medicine assumed they meant. The sickle cell variant became common in West African populations because having one copy of the mutated gene provides significant resistance to malaria, which was, and in some areas remains, a major cause of death. Natural selection favored that variant in a malaria endemic environment, regardless of racial category. A person from Southern Europe or the Middle East living in a historically malaria-prone region might carry the same variant. Two people classified as the same race, but with ancestors from regions with very different disease histories, might have completely different risk profiles. Using racial category as a proxy for genetic disease, risk produces less accurate medicine than using actual genetic information about the individual patient. The genetic data is specific. The racial category is an approximation that sometimes happens to be useful and sometimes fails entirely. The concept of race as a biological category emerged in European scientific writing during the 1700s and 1800s. Scientists of that era tried to classify human diversity, the same way botanists classified plants and zoologists classified animals. They created hierarchies. They ranked populations in order of supposed capability and worth. They used those rankings to provide scientific cover for colonialism and slavery. They looked at differences in technology, social organization, and cultural practices between populations, and attributed those differences to innate biological characteristics, rather than to historical circumstance, geography, and the compounding effects of time. Those early racial scientists genuinely believed they were being objective, but their conclusions reflected the power structures of their time more than the actual data available to them. And the genetic data collected over the last 30 years has systematically dismantled every specific claim they made. The traits those scientists use to rank populations have no measurable correlation with intelligence, judgment, creativity, or any other cognitive capacity. The 0.1% of the genome that differs between individuals is distributed randomly across populations for traits that matter for those capacities. You cannot predict a person's cognitive ability from the genetics of their skin color, any more than you could predict the speed of a car from the color of its paint. The traits are unrelated. The data produced by comparing the DNA of populations across the world does not support racial hierarchy. It does not support racial purity. It does not support racial separatism. What it supports is a picture of one species with a recent common origin, massive within population variation, relatively small between population variation, and a history of movement, mixing, and mutual influence that has been continuous since before anyone had names for any of the places involved. The disparities in wealth, health, education, and opportunity that exist between groups defined by racial categories in many countries are not reflections of genetic differences between those groups. They are the products of specific historical decisions, specific systems, and specific ongoing practices. The genetic evidence makes this clear by eliminating the only alternative explanation. There is no genetic foundation for those disparities. They must therefore have other causes. And causes that are not genetic can, in principle, be changed. The scientists who built the database and ran the analysis did not set out to make a political statement. They set out to answer a scientific question about the nature of human genetic variation. The answer they found is what it is because the data is what it is. And what the data shows is that the human family is far more unified, far more intertwined, and far more recently connected than centuries of classification suggested. The visible differences between us are real but shallow, recent but not ancient, adaptive but not fundamental. The deeper genetics tells a story of shared origin, continuous mixing, and biological kinship that no political or cultural system ever fully reflected. And what we choose to do with that knowledge is the question that the data cannot answer for us. Subscribe now if you want to keep following science like this, the kind that actually changes how we understand ourselves. Drop a comment below with your reaction to the most surprising finding in this video, and share this with someone who should know what the largest genetic study of human diversity actually found. Thanks for watching. Let us go deeper into one specific area of this science that has the most direct real-world consequences, because it is where the gap between what the genetics shows and what some current practices assume is the largest. Medical research for decades used racial categories as shortcuts. If a drug was tested primarily on one group and produced certain side effects at certain rates in that group, those statistics were often applied to other groups, classified as the same race, without checking whether the underlying genetics actually supported that assumption. If a disease showed higher rates in one racial population, researchers sometimes assumed that meant a biological predisposition inherent to that race, rather than asking whether the patients in that category shared specific genetic variants, specific environmental exposures, specific historical experiences with healthcare systems, or some combination of all three. The genetics data challenges every one of those shortcuts. Genetic variants that affect how a person metabolizes a particular drug can be present in some members of a population and absent in others within the same population. If that variant happens to be more common in population A than in population B, that is useful information for statistical purposes at the population level. But applying that population level statistic to an individual patient without testing that individual is less accurate than testing the individual directly. And the categories used to assign people to population A or population B in the first place, the visible racial categories, often do not align well with the actual genetic variation underneath. Two patients who look the same, who identify with the same racial group, who would be checked into the same racial category on a medical form, might have completely different genetic profiles for the variants that matter for their treatment. And two patients who look different, who identify with different racial groups, might share the genetic variants that matter. This is not an abstract concern. There are documented cases of medical errors caused by applying racial statistics to individual patients whose actual genetics did not match what those statistics assumed. And there are cases where patients belonging to populations that were underrepresented in research cohorts received treatment calibrated to the genetic profiles of different populations. The solution? The genetics research points toward is direct. Test the individual. The technology to identify the specific genetic variants that matter for drug metabolism, a disease risk, and treatment response exists and is increasingly affordable. Using it produces more accurate medicine than any racial category can. There is also something worth saying about what the data does not show, because a careful statement of the limits of the findings is part of what makes the science honest. The genetic research does not show that all humans are genetically identical in every way that matters. The 0.1% variation is real, and some of that variation has real effects. Different populations do have different average frequencies of specific genetic variants, and some of those variants do affect health outcomes in measurable ways. The genetics does not erase those differences. What it shows is that the traditional racial categories, the 5 or 6 or 8 boxes that racial classification systems typically use, are not the right unit for thinking about those differences. The variation that actually matters for medicine, for pharmacology, for disease risk, does not align with the visible categories. It aligns with specific genetic variants that can only be identified through direct testing. The genetics also does not show that culture, language, geography, and shared history are not real or important. They are. The populations that the Human Genome Diversity Project sampled are real communities with real shared experiences, real languages, real cultural practices, and real histories. Those things are important and meaningful. What they are not is the same thing as genetic groups. Cultural and linguistic communities often have shared genetic ancestry, but the correspondence is imperfect and has never been perfect. Populations have always mixed. Languages have always spread beyond the communities that originally spoke them. Cultural practices have always moved between groups through trade, migration, and contact. The idea that culture, genetics, and geography perfectly overlap to define bounded racial groups was always an oversimplification, and the genetic data makes that oversimplification impossible to maintain. The Human Genome Diversity Project was followed by larger and more detailed studies, including the 1000 Genomes Project, which sequenced complete genomes from populations across multiple continents, and the work of the Allen Ancient DNA Resource, which has been sequencing DNA from thousands of ancient human remains to trace population movements across deep time. Each new dataset has confirmed and extended the same basic findings. African populations are the most genetically diverse. Genetic diversity decreases with distance from Africa. Visible traits evolved recently and do not track overall genetic relatedness. All modern populations are mixtures. The concept of biological race does not find support in the data. What those studies have added is resolution and detail. We can now trace specific population movements with remarkable precision. We know that the people who built Stonehenge were largely replaced by incoming Bronze Age migrants from the steppe. We know that the ancestors of modern Japanese arrived in waves from the Korean peninsula and mixed with the indigenous Jomon population that had inhabited the Japanese islands for thousands of years before. We know that the ancestors of the Polynesian populations who colonized the Pacific Islands, reaching Hawaii and New Zealand and Easter Island in extraordinary navigational feats, were themselves a mixture of Austronesian seafarers and populations from Papua New Guinea. We know that the Inuit populations of the Arctic represent a relatively recent migration into North America that is genetically distinct from the earlier migrations that populated the rest of the Americas. The human story, read through DNA, is a story of constant movement, constant mixing, and continuous change. The researchers who put the Human Genome Diversity Project together did not know all of this when they started. They knew the question they wanted to answer. What does human genetic diversity actually look like? The answer they found was not the answer that centuries of human classification had assumed. It was more interesting, more complicated, more unified, and more surprising than that. And it replaced a set of assumptions built on visible differences with a picture built on actual data. That picture shows a single species with a deep shared history, recent surface-level differences that evolve for good functional reasons, no meaningful genetic boundaries between populations that would justify the categories our social systems have treated as fundamental and a degree of interconnection. Going back through family trees and ancient migrations and unexpected contacts across vast distances, that makes the boundaries we draw look fragile and arbitrary against the actual record of how our species has moved and mixed across the planet. One species, one genome, one family with a recent common ancestor and a long common future. That is what the largest genetic study of human diversity ever conducted actually found. If this kind of science is what you come here for, subscribe now and turn on notifications. Drop a comment below with the finding that surprised you most. And share this with someone who should know what the data actually shows. Thanks for watching.